So Paige, you got the email from Angie, correct, about her not getting it this week?
So
That's when I told you I'd take everything from here on out.
or we could-
Paige.
I got you.
Great, all right, well, this is really an amazing technical feat for us to try to pull this off, both hybrid and so we have folks here in G217, including our visiting professor and hopefully people that are logged in and will come in and out over the course of the afternoon during these presentations. As you know, this is our 29th annual Charles Huggins Research Symposium. And we're really thrilled that last year was entirely virtual. And this year, at least, we're back to some semblance of hybrid. Huh? How's that?
It might be off.
Is the mic not on?
Try it again.
No, it's on.
No, he said, I think I see a thumbs up from John.
Can you
John
hear
is
me?
giving you a thumbs up.
Is that good?
Yeah, everything's fine. I can hear you perfectly, Jeff.
Oh, Paige was yelling at me. What you need or something? Okay, so anyway, we are going to get started here. We've got a really exciting program. It's always one of the highlights of the year, the Huggins Symposium, where we can really celebrate the legacy of Dr. Huggins, as well as our scholarship that derives from our department in so many exciting different ways. And it's only a small little soup song of the work that goes on over the course of an afternoon. We can't, of course, capture all the exciting work that's done, but at least we can showcase some of it. Here we go. As you know, Charles Huggins was born in 1901 in Halifax, Nova Scotia, and he passed away in 1997 here in Chicago. He was on the faculty of the University of Chicago for 45 years. In addition to the Nobel Prize in Medicine in 1966, over 100 awards and honorary degrees, and really is the person who sort of established under Dr. Femister's leadership the notion and the importance that research by surgeons mattered. It had impact, and his legacy is really quite incredible. You know that in 1966, his work for discoveries concerning hormonal treatment of prostate cancer led to a series of 3 papers showing dramatic responses of prostate cancer to castration. And his subsequent investigations into oophorectomy, adrenalectomy for breast cancer, established the response of certain tumors to hormonal manipulation, but really introduced the concept of fingerprinting the tumor and targeting therapy for it here based on hormones, but obviously his work led to the establishment of the Ben Cancer Research Institute here. His legacy in individuals like Janet Rowley and others, all the work that here that led to the discovery of the estrogen receptor, and that is targeted therapy as well. It was inspired by his work and really generations of surgeon scientists, but as well as full-time scientists derived from his influence. He shared the Nobel Prize with Peyton Rouse of the Rouse sarcoma virus, and really ushered in that era of rational chemotherapy and personalized medicine. So the schedule of events for today is the following. I'm going to announce the winners of the posters that have been up online for the last couple of days based on your votes. We will then have selected presentations for the winners of the faculty, postdoc, resident, and student categories. Then we will have a series of 4 presentations by 4 of the senior research, the second year research residents in the lab now. We'll have some quick shots from the poster winners that I'll announce. Presentation from the education and DEI categories. And then at 4 p.m., we're really excited to have Dr. Saray Parengi, who's a professor of surgery. I will do a more formal introduction later. Professor of surgery at Harvard and well-known, busy endocrine surgeon, surgeon scientist at the Mass General, and chair of surgery at the affiliated New Wellesley Hospital. And she is giving us a talk at 4 p.m. on hope and resilience, which I can assure you will be spectacular. So without further ado, let me announce the poster winners. The best poster winners who we will hear from later are Ashley Diaz from Dr. Elizabeth Blair's lab, Dr. Jack Kent from Lucia Madriaga's lab, and Jared Pollard from Anita Chong's lab. So congratulations to all of you. Also notable that we had a hat trick out of the Pachavasek lab for best image, best poster explanation, and most exciting innovation. So the hat trick won by Kathleen Cow, Camille Cabaz, and Nong Nguyen. So congratulations for really presenting some very, very exciting work. Your moderator for today's activities will be the eminent Dr. John Alverdi, zooming in from home, and he will be announcing the speakers. Please, we'll be watching the chat and the Q&A. Ask questions. We can also give you microphone functions to ask questions if you're from Zoom, and we have our little catch box here so we can ask questions that anybody can hear. We'd like to get some good discussion going about these papers because often it leads to new collaborations and new ideas that come out every year as well. So let me turn it over to you, Dr. Alverdi, and you're on.
Well, thank you, Dr. Matthews. It's always a pleasure to hear about Dr. Huggins, and he's such an inspiring figure and has such a strong legacy here at the University of Chicago that if anyone has a chance to talk to more senior faculty about his legacy, he was not only a brilliant researcher, but an interesting character with lots of very interesting stories from a number of senior faculty here at the University of Chicago. Unfortunately, our first speaker, which is on the program, Dr. Perez Gutierrez, is not available. So we're going to move right to Dr. Dima to discuss the role of tumor-informed personalized ctDNA assays in informing recurrence in patients with peritoneal metastases from colorectal and high-grade appendix cancer undergoing curative surgery from the Kiran Taraga lab. Dr. Diamond.
Thank you very much for allowing me today to present my research. I am going to discuss the role of tumor-informed personalized ctDNA assay in informing recurrence in patients with peritoneal metastasis from colorectal and high-grade appendiceal cancers who underwent curative resection. These are our disclosures. Peritoneal metastasis is traditionally considered as a lethal endpoint for various intra-abdominal malignancies. Even after optimal cytoreductive surgeries, high rates of peritoneal recurrences are observed, around 50% at 1 year and 70% at 3 years. The conventional tumor markers such as CEA have shown limited clinical utility in terms of detecting these recurrences. Moreover, 50% of these recurrences were undetectable by routine imaging investigations. Therefore, better prognostic markers are urgently needed to improve the patient outcomes. Circulating tumor DNA or ctDNA is rapidly emerging as a non-invasive diagnostic and prognostic biomarkers. Studies have shown their clinical validity in terms of early detection of cancer, risk stratification, as well as monitoring response to therapy. However, their precise role in peritoneal metastasis is yet to be defined. Therefore, we investigated the role of ctDNA in informing recurrence in patients with peritoneal metastasis who underwent curative research. We utilize Signatera, which is commercially available tumor-informed next-generation sequencing-based multiplex PCR ctDNA assay. And as I said, it is personalized and tumor-informed. We first take the tumor tissue, analyze it using next-generation sequencing to identify tumor-specific mutations, and then test these mutations using PCR in the peripheral blood. Studies have shown its superior performance over conventional distal drop PCR-based ctDNA assay in predicting recurrence in metastatic colorectal cancers after surgery. The methods. The single institution study was conducted at the University of Chicago Medical Center. Our inclusion criteria were patients with colorectal and high-grade appendix cancers with peritoneal metastasis who underwent curative resection between 2018 to 2021 and had serial postoperative ctDNA assessment with signature. We excluded patients who had suboptimal cytoreduction, have positive ctDNA immediately after surgery. So we specifically excluded those patients who had positive ctDNA. We only included those after surgery who had undetectable ctDNA. And then who had follow-up less than 6 months and extraperitoneal metastasis at the time of surgery. Here is our study schema. These patients with peritoneal metastasis from high-grade histologies, they first undergo diagnostic laparoscopy to assess the disease extent, followed by 3 to 6 months of systemic chemotherapy. Then they have cytoreductive surgery with or without HIPEC, and they complete their 12 cycles of chemotherapy, not completed before surgery. We tested ctDNA after 4 to 6 weeks of surgery, and then followed these patients 3 monthly with ctDNA, conventional tumor markers, and routine cross-sectional imaging. Our primary outcomes were recurrence rates and disease-free survival, and here are our secondary outcomes. The results. Here is the console flowchart of our study. We finally included 33 patients, of which 13 were colorectal cancer and 20 of appendix cancer. The comparison groups were rising ctDNA cohort with 19 patients and undetectable ctDNA cohort with 14 patients. The baseline characteristics such as demographics, they were well balanced between the two groups. Similarly, the tumor characteristics, whether the patients had appendix cancer, colorectal cancer, synchronous or metachronous peritoneal metastasis, as well as the PCA score, they were well balanced. And finally, the pathological characteristics such as pathological grade or presence of signature cells, they were again well matched. As we can see here, 90% of the patients in rising ctDNA cohort had recurrence, compared to only 21% in the patients who had undetectable ctDNA levels. And this was statistically significant. Our median disease-free survival for the whole cohort was 12 months over a median follow-up of 13 months. And as we can see in this Kaplan-Meier curve, the disease-free survival was markedly reduced for the patients with rising ctDNA, with a median disease-free survival of 11 months. Compared to it was not reached in the patients with undetectable ctDNA, and it was statistically significant with a log-rank p-value of 0.01. We further tested the various clinical pathological factors in a POX model, and on univariate analysis, rising ctDNA was the single most important prognostic factor associated with disease-free survival. After multivariate adjustment, it holds its significance with a hazard ratio of 3.67. In our cohort, the median overall survival was not reached as only 3 patients have died so far. In our study, the sensitivity and specificity of ctDNA to detect recurrence was around 85%. And as we can see here, the ctDNA was able to predict recurrence in most of the patients way before it actually appeared on the radiographic imaging. So our median ctDNA lead time was 3 months. When comparing with CEA levels in those patients who had recurrence, ctDNA outperformed CEA levels with a sensitivity of 85% compared to 50% sensitivity of CEA levels. Concluding, our study results support the clinical validity of serial ctDNA assessments as a strong prognostic biomarkers to detect recurrence following curative intent resection in patients with peritoneal metastasis. It further promises for informing future clinical trial designs to test early and aggressive therapeutic interventions in patients who are ctDNA positive but not have clinically progressed. Also, the next generation sequencing-based ctDNA assays may help us identify targetable mutations in treatment-resistant cases. Our study had limitations as we have small sample size and was retrospective design. We also combined 2 primaries, which may affect the generalizability of the results. And further, the study design does not establish any changes in treatment strategies for those becoming ctDNA positive after curative intent surgery. As a note to further research, we are enrolling patients in a prospective clinical trial where we are studying 5-hydroxymethylation signatures in plasma cell-free DNA as a marker for tumor burden and recurrence in appendix and colorectal peritoneal metastasis patients. 5-HMC or 5-hydroxymethylcytosine is a promising epigenetic marker and is molecularly very stable, and it signifies gene expression. We are using a novel nano-HMC seed technology, which was developed at the University of Chicago, and is highly sensitive and robust method to profile 5-HNC in as low as 5 nanogram of DNA, making it very suitable for use in cell-free DNA in peripheral blood. As we aim to discover epigenetic signatures for peritoneal metastasis, our preliminary results were very promising. As you can see here in this PCA plot and heat map, we were able to distinguish patients with peritoneal disease with those without peritoneal disease. And this prospective clinical trial design is powered to assess the clinical validity of plasma 5-HNC signatures in identifying the clinically detectable recurrence at the 1 year of surgery. I would like to thank my mentors and my collaborators, as well as the Department of Surgery, University of Chicago, for giving me an opportunity to be a part of this amazing culture. I would like to thank you all again for your time, and I welcome any questions.
Thank you very much, Dr. Daimar. Are there any questions? If not, I'll start it off. If you have a question, please let Ms. Kirkpatrick know, and she will release you as a host, and you can speak directly. I'll start off the question. Great presentation, nice job. Thank you very much. Can you just outline, I sort of missed it at the beginning, about how the tumor ctDNA, or how you sequenced the tumor itself to arrive at a blood test that had enough stringency to it that it was sensitive. And I fully appreciate how you timestamp these samples so that, you know, they were more predictive than would be, let's say, imaging studies or recurrences that occurred clinically. Thank you.
That's a good question, Dr. Alberti. And I would like to say that it is a commercially available test where we first test the tumor tissue. As we do laparoscopy for all the patients, we take tumor specimens. And then we send these tumor tissue for next generation sequencing to the lab. And then they test for specific mutation, mainly 16 cancer-related mutations, which are highly important for colorectal cancers, for example. And then they specifically see those tumor-specific mutations in the peripheral blood using multiplex PCR.
Yes, Dr. Shogan has a question. Can you release him, Paige, so we can hear him? Go ahead. Go ahead, Ben.
Hey, it's Ben Shogan. Great talk. One thing that I pick up on, was it that 21% or so of patients that end up found to have a recurrence had a non-detectable rising ctDNA?
Yes.
And at what time point was that after their most recent HIPEC? Was it just that it was too early, or was there a time, you know, a longitudinal time issue with that, or what do you think the reason is that that was such a high number?
I would say that all of the patients had a median follow-up of around 13 months. And I would say that most of these cases which recurred, even if they were ctDNA negative, they recurred within that 1 year period of time. And I would like to again say that there is another limitation of our study, that we didn't test ctDNA before surgery. So we didn't know that whether the ctDNA was actually coming positive or was actually sensitive for these patients before surgery as well. So we are conducting this clinical trial in which we are assessing ctDNA before surgery and then serial ctDNA assessment after surgery.
Oh, thanks.
Dr. Brandy.
Great presentation, very interesting area of work. I have a couple questions. One was, so your original assay, you showed as it was taking the tumor from the colorectal location. So do you think that in most cases, people won't have that benefit, right? Because they get the recurrence already, like they're not at a place like University of Chicago where this kind of assay would be used early. What do you think the utility will be if you don't have the primary tumor specimen? How important will it be to get that? Like, should you do laparoscopy just to get that, to get the most benefit, for example?
So I would say that we studied these tumor mutations or next generation sequencing was done in the metastatic tissue per se, because we generally get those patients who are already operated for primary cancer and then
That's gone.
detected with peritoneal metastasis. And for primary colorectal cancer, we can take the tumor tissue while doing colposcopic biopsies, and then use those tumor tissues to detect these tumor-specific mutations. However, this is a good question, and I would like to say that tumor architecture or tumor mutation profile do change because of various therapeutic interventions. And that's why, is it specifically the presence of DNA in the blood or these mutations is still unknown. So that's why we are conducting our own signature, trying to discover our own signatures in the peripheral blood so that we can say that, okay, these mutations are important and we should test these mutations in future.
And I have two other questions. One is, did you find that it was tumor burden dependent? So like the bigger the tumor burden, the bigger your signal, or not really?
Not really, because we adjusted the median PCI score as well as we adjusted, as we have seen that the PCI scores were well balanced between the two groups. So, and also I would like to say here that the peritoneal metastasis is well known to shed very less ctDNA in the peripheral blood. And when it was positive for peritoneal only recurrence, it was like very slightly positive. I would say like, as compared to liver metastasis recurrences, it was very, very, very low.
Oh, nice. Very interesting. And then what do you think you can do in that lead time? Like what benefit will that give the patient? So now you showed this, impressive lead time. I mean, 3 months. What can you do with that lead time before it shows up in imaging? Like what are
we
things?
can follow the patients aggressively, and if we can detect, for example, if we can detect peritoneal recurrence early at a stage where we can do another CRS or another CRS IPEC, then I think those can benefit the patients. However, in a clinical trial, it was shown that it didn't benefit the patients, but it's still a topic where the research is still going on, and I would say that benefit should be given to the patients.
It's interesting because we're seeing patients with lots of different types of tumors now getting ctDNA, measured, and we don't really know what to do with the information because we don't have well-designed trials to know whether the lead time of recognition of how predictive it is of the fact that metastases or recurrence is going to occur, or then to Dr. Prangi's question about whether you can do anything about it. So it's a difficult situation. It's, I think, what Dr. Alberti calls guilty knowledge. You have knowledge of things that you would rather not have of known, because all it does is take away hope for the patients, a month or two sooner than they would otherwise have, or maybe a year or so sooner. So it's hard to know what to do with this, but it's an incredibly hot topic and it's really interesting research. Are there any other questions that we have here? Mitch or Carrie, did you want to make any comments?
I don't have any.
You actually asked the question, I'm sorry,
in the Q&A. Sorry.
you asked the questions that was really the lead time issue, because the real question comes is that, and I've never gotten the answer myself, is whether picking up recurrence early has value, or is it just a biologic marker? And the question comes up, and this was also raised by our keynote speaker, is it a reflection of tumor burden, or is it a reflection of something else that we can intervene on? And I do think that the anxiety of a elevated ctDNA is extraordinary, and we don't know what it all means, actually. And I think that as you design your studies, I think it's going to be really important to determine whether earlier intervention really has value. And that's going to be hard to show, actually.
I agree with you, Dr. Pooner, that this could only be answered in a setting of a clinical trial where we can actually offer early and aggressive treatments to these patients and see whether it actually makes any difference.
Yeah, and I would applaud you because, as you know, the literature on peritoneal malignancies is without clinical trials, essentially. We just base it on a gut feeling. So what you're doing is actually unique, and your presentation was just fantastic, just so you know.
Yeah, I agree.
Thanks, Mitch.
Great
Oh,
job.
Michael.
And,
Do you want to-
I will say, as an aging male who, contemplates when and how to get their PSA checked, and this is interesting, especially in view of all of Dr. Huggins' work, that PSA was one of the early biomarkers that spills into the blood so easily, it's easily detectable. And yet we're now still to this day contemplating, to Dr. Poizner's point, and Dr. Matthews' point, when or if to get a PSA on patients. And so this sort of falls right back into that same sphere of influence in terms of patient anxiety and whether we can act on information that we generate. Very interesting.
Dr. Trago wanted to make a comment.
Yeah,
Oh,
I think,
sorry.
obviously, Ankit, you did a very nice job, and thanks for that presentation. This is fascinating for us because the biggest problem we see in patients with peritoneal metastases is metachronous recurrences after colon primaries resected. Patients are doing fine, they live a normal life, and then suddenly they have peritoneal disease and they're in your ER with bowel obstructions dying. And so I think it's a nice way for us to find these tumors, but I think we're really struggling to figure out whether we should use the tumor-informed DNA, which is more sensitive, but because DNA is shed at such low levels, is it better to just get cell-free DNA? So just kind of search in the DNA itself along time points to see if the mutational profiles are changing. And the methylation signatures that we're looking at, they're actually very cheap. So ultimately, we think when they're commercialized, they'll cost $90 compared to, say, the tumor-informed DNA assay that we have here, which is like $20,000, $30,000. And I think what is also interesting is that a lot of trials are now being designed not only just for detection and what you do, but the therapeutics. So like in our trial, in this study, for instance, if they're recurring within a year, it's unlikely we're going to offer them another surgical intervention. But you can actually use different chemotherapeutic regimens and more molecularly targeted agents based on the DNA profile. So, I mean, this is all conceptual and theoretical, but I think it goes to the concept of personalized medicine, a little bit like what Dr. Huggins did is like, how do you actually get to sort of what the disease is and potentially try to target the disease more as opposed to just a blanket sort of treatment of FOLFOX or FOLFIRI or something like that. So yeah, exciting, but again, lots of more questions.
Know when to
More
use
questions,
those,
less answers.
know when to use those mitomycin labs.
Yeah.
Coming back to that.
It always circles back to that.
Never live that down.
Okay, let's go to our next presentation. Remember that each and every one of these presentations represents somebody who scored very high by our referee panel, and each and every one of them are award winners. Our next up, along the same theme of metastasis, is Dr. Ryan Morgan, who is from Ben Shogan's lab, and he's going to speak to us on the development of postoperative local tumors and distant metastases is diet, genetics, and microbiome dependent in a mouse model of colorectal cancer recurrence.
If I can also give Dr. Morgan a quick shout out and thank you as our on-site technical person who has helped us out greatly this year so far. So kudos and thank you so much for that as well.
Crash course in Zoom this year. Thank you, Dr. Alberti, and thank you to the department for the opportunity to share our work from the Shogun lab. Colorectal cancer is the third most common cancer in the United States, and it remains one of the leading causes of cancer death. Many patients can be cured with resection, but there's a significant proportion experience postoperative recurrences. The risk of recurrence is proportional to the patient's stage. However, even patients with early stage disease and negative resection margins can experience a recurrence. Given the severity and frequency of these events, there's a pressing need to further understand the mechanism underlying this. Based on previous results in our lab and the Alberti lab, we have developed a new model of postoperative colorectal cancer recurrence. In addition to the primary tumor at the time of surgery, there are viable shed tumor cells within the lumen of the bowel. When the primary tumor is resected, these cells remain within the lumen of the bowel and are able to interact with the anastomotic environment. Multiple factors can impact this environment, including diet, particularly a high-fat diet, which increases cancer risk, microbiota around the anastomosis and the post-surgical changes, and also tumor genetics. We propose that by altering and interacting with the microenvironment, these factors can promote the migration of these intraluminal cells through the healing anastomosis to cause local recurrences of distant metastases. Our hypothesis in the study was that a high-fat diet and the associated changes in the microbiome will influence the anastomotic microenvironment and invariably promote the migration of these intraluminal cells based on their genetic profile. To mimic these intraluminal cells, we've been using colon cancer organoids. These are three-dimensional cancer cell cultures with multiple cell types, and compared to a monolayer culture, they can somewhat better recapitulate the tumor microenvironment. Specific to this study, we're able to use organoids with colorectal cancer driver mutations. In prior models, different tumoroids with different genetic backgrounds have demonstrated a wide range of metastatic potential, ranging from limited disease to overwhelming hepatic and pulmonary metastases. However, most of these models have been either in primary mice or in splenic injection models. And as surgeons, we wanted to introduce these tumors to a model that more closely resembles recurrence after surgery, and in which we could see how this metastatic potential responds to changes in diet and the microbiome. We're able to use two different organoids for our study. The first is AKPT. In primary mice, this models the adenocarcinoma pathway of colorectal cancer pathogenesis. It's an APC, KRAS, P53, and TGF-beta1 receptor mutant. Second organoid we used was KPN, which models a serrated pathway in primary mice. This is a KRAS, P53, Notch mutant. And based on prior models, we would have expected KPN to have a more aggressive phenotype in our model. To study this question, we used a model that was first described by Dr. Gaines during her time in the lab. Mice are given either a high-fat diet, which is 60% calories from fat, or a normal chow diet for 6 weeks preoperatively. We then create a colon anastomosis, and on postoperative day 2, we give mice a cancer cell enema with either AKPT or KPN cells to mimic those intraluminal cancer cells present at the time of surgery. We then serially sacrifice the mice, focusing on postoperative days 21 and 56, and then collect samples to assess for tumor formation. We first sought out to show that these organoids can cause both local and distant tumors in our model. Here we see AKPT tumors, both at the anastomosis in the colon and distantly in the liver and the lung, on both gross appearance and H&E staining. We then confirmed that these are AKPT cells within these tumors by performing in situ hybridization for LGR5, which is a colon cancer or a colon stem cell marker. We then repeated these experiments for KPN, and we're able to show that both of these organoids can cause both local and distant tumors within our model. When we looked initially at those two main time points of day 21 and day 56, comparing mice and both organoids with either a chow diet or a high-fat diet, the rates of tumor formation were variable. However, there was this marked increase in tumor formation in mice given an AKPT tumoroid at day 56 when they were given a high-fat diet. And including some other time points that aren't shown here, we noticed a consistent pattern where these mice that had an AKPT tumoroid were given a high-fat diet had earlier, more frequent, and bulkier tumor development compared to KPN mice that were given a high-fat diet. We also noticed subjectively that the AKPT mice seemed to be dying earlier, not reaching their endpoint. We're able to show that there was a significant decrease in survival for mice given an AKPT enema when they were put on a high-fat diet rather than a chow diet. And interestingly, we did not see the same effect in KPN organoids, suggesting that this negative response to a high-fat diet is dependent on tumor genetics. Having shown that a high-fat diet leads to a more tumorigenic and lethal phenotype in AKPT mice, we then wanted to see whether we could reverse this phenotypic effect by changing the mice's diet preoperatively. In a repeat set of experiments, we again had a chow group of mice fed just a chow diet, a high-fat diet group, and then a reversal group. And in this group, we gave mice a high-fat diet for 6 weeks preoperatively, followed by a reversal period with a chow diet for 2 to 6 weeks. We then performed our anastomotic model and sacrificed on postoperative day 56. When we started, we used a 2-week reversal period, and what we noted was that the reversal group continued to have increased mortality and high tumor formation rates similar to the high-fat diet mice. However, when we extended that reversal period to 6 weeks, we noted that the mice had significantly improved survival within the reversal group and had low tumor formation rates similar to the chow-fed mice. We hypothesized that this difference we were seeing with the extension of the reversal period was due to a time-dependent change in the colon microenvironment that these cells were interacting with. And then we sought to assess the mechanism. The first step we took was to perform 16S analysis to look at the microbial composition at the site of the anastomosis at serial time points in all the groups in the previously mentioned reversal experiment. In the high fat diet group, we noticed most particularly that there was a high concentration of allobaculum, shown here in orange, on postoperative day 2 that persisted to postoperative day 0 at their baseline, which persisted to postoperative day 2. We then noticed on postoperative day 7, there was a bloom of Enterococcus, shown in green, as well as Proteus, E. coli, Enterobacter, and Shigella, shown in red. The reversal group mice had a significantly different microbiome compared to the high-fat diet mice. They had increased levels of Bacteroides and Lactobacillus at baseline, shown here in teal and blue respectively, a decreased allobaculum, and then the most marked change we saw for them was again an Enterococcus bloom that came earlier on postoperative day 2, and there was no commensurate increase in E. coli, Proteus, or Shigella. Most importantly, what we found was that the baseline microbiome in the reversal group and the response to surgery very closely mirrored what we saw in the chow diet mice and was significantly different than the high-fat diet mice. Having shown that the high-fat diet-associated microbiome was distinct from the chow diet, we wanted to then isolate its effect in germ-free mice. In these experiments, germ-free mice were given a normal chow diet, and we had one group that underwent our anastomotic model with no preoperative intervention, and a second group that received a fecal microbiota transplant from high-fat diet conventional mice. We then sacrificed the mice on postoperative day 56 to assess their tumor formation. There's a third group with a chow diet FMT, which is ongoing. What we found was that when isolated in a germ-free mouse model, the microbiota associated with the high-fat diet were able to promote AKPT tumorigenesis. In our control germ-free group, there's a low 10% rate of tumor formation, and most of the anastomoses remain disease-free, as shown on the left here in blue. However, when we introduced the microbiota from high-fat diet mice, we saw a significantly increased 40% rate of tumor formation, conferring almost exactly the same phenotype as we saw in the conventional high-fat diet mice. So in summary, so far, we've been able to show that both AKPT and KPN tumors can form both local and distant tumors in our model. However, AKPT forms tumors earlier and more frequently. A high-fat diet, specifically with AKPT, increases tumor burden and decreases survival, suggesting that the response to diet is based on tumor genetics. This high-fat diet-induced tumorigenesis can be attenuated with diet manipulation and also corresponds to a reversal of the microbiome. And when we introduce this microbiome to germ-free mice, it can confer a highly penetrated tumor-forming phenotype in these germ-free mice that normally would not form tumors. The next steps for our group will be to further assess the mechanism underlying this. Ongoing experiments include functional microbiome analyses, RNA sequencing, and metabolomics, which we expect to have back in the next couple of months. I'd like to thank ASCARS for supporting this project with the General Surgery Resident Research Initiation Grant. I'd also like to thank Dr. Shogun, everybody in the Shogun lab, as well as all of our collaborators across multiple different departments in the university. And lastly, I'd like to thank Dr. Matthews and the rest of the leadership in the Department of Surgery for allowing me the time to take 2 years in the lab and for the continued support of this project. Thank you all for your time.
Great job, Morgan. Congratulations. I think there's some questions. Dr. Chong, do you have a question? Can you put her on page so that she can speak? Is Dr. Chong in the audience there, or is she remote? I don't see her.
Page is here. We have to get Dr. Chong promoted.
Okay.
We can do it from here. Hang on a sec.
Okay.
Hang on, Anita. Oh, it's written in the chat there.
Yeah.
So the question is, does changing the diet back, I don't know if she wants to ask it live, but changing the diet back to normal chow restore the anti-tumor response?
So we, for those reversal experiments, it seemed to be time dependent. However, when we extended that out to 6 weeks, the only real evidence of what we have of what changed so far is the analysis of the microbiome, the composition changes after the reversal period. And in some way that is related to improvement in the anti-tumor effect. I think, as of right now, the question is still for us what the exact mechanism is for that, but we are starting with the microbiome analysis.
Ryan, the setup here is that somehow the anastomosis itself is a weak point, that's a potential place of entry. How dependent on your results is that hypothesis really? Because somehow I struggle with the idea that there's so many tumor cells mysteriously floating around in that luminal environment post-operatively, that's the path, where the local recurrence, happens versus, multiple potential routes. I mean, how does it get to the liver and other places? I just have a hard time believing it's through the anastomotic wound there. The other question is, in mice where you can control things, it's great because we can do experiments and we can get an outcome. Integrating that back to the human situation where people, have bowel preps, they have antibiotic or not antibiotic. It's a super complicated clinical situation. How are you going to dissect out the multiple changes of somebody's microbiome and the week before they have their operation, their bowel prep, their antibiotic treatment, et cetera, try to figure out how big a signal this would be towards recurrences in humans? Great job.
To your first question, one thing I didn't have as many photos in this presentation. However, every mouse that I've operated on over the last 2 years that has developed a tumor with this model, the tumor has been right at the anastomosis, which I think is fascinating because we do have, in the sense of an internal control that mice all have these internal cells and they only form tumors at the anastomosis. The other thing we've noted is that when we take histology pathology right at the anastomosis, the tumors tend to be extra luminous. We're not seeing coastal tumors, we're seeing them all on the serosal side of the bowel, which I think is consistent, but it doesn't necessarily prove that the cells are migrating through the anastomosis. But
it just means maybe that they land there from wherever they come from, because maybe there's a wound there that's, that's how they reach it, I suppose.
Yeah. And to your second question, I agree. I think that in our patient population won't be so clean. However, we do have some evidence. Dr. Zhang and Dr. Sherman had a paper last year that if patients receive an antibiotic preparation, combined mechanical and antibiotic preparation, there's about a 10% increase in 5-year disease-free survival compared to those patients that don't receive a bowel prep. So I think that it wouldn't be it would be a single thing that really improves outcomes, but I think things like the antibiotic preparation and some things we'll find here, we can slightly improve those outcomes. Great.
I wonder about working with, you have the ultimate recurrence model, which is Dr. Taraga's patients. So maybe there's a way to sort of bring those together.
Ryan, great presentation. This is a piggyback onto Dr. Matthews' question, because and I'm sure it's because I'm just not smart enough to figure it out, but I struggle with the model in the sense that very few patients have especially with colon cancer, have a local recurrence. And there's a lot of the colon patients who obviously have distant disease. So I struggle with the idea that this is somehow the cells are going through the anastomosis and eventually they get into the bloodstream or whatever. But how many of your mice get a distant metastases without local recurrence?
So that's definitely a limitation of this model. That for all the mice that have distant recurrences, they also have a local tumor within this model. It's fairly reliably in the liver and the lungs, but we don't see mice with, for example, large liver tumors in the absence of a tumor at the anastomosis. And I agree that is a limitation in that a lot of the patients we see will present with distant metastases.
And the other component, even with rectal cancer, it's been shown by Professor Heald's study that, surgical technique actually leads to decreased local recurrence even. It's just something I struggle with, but, the work is so elegant that it still, it opens doors to thinking about how the microbiome has an effect on distant disease and local recurrence. But I just struggle with the model a little bit.
That's something that we discuss in the meetings as well.
So Ryan, we've also been looking at the metabolomic pathways. And the question is is the microbiome a bystander, or is there more to this? And we've seen 2-3 things. So we've seen that the lactate levels are changing, and they change the macrophages from type 1 to type 2, and that's sort of causing more. In our plasma signals that Ankit presented earlier, we saw overexpression of glutamate genes. And so it's very fascinating just sort of looking at what happens, and especially for peritoneal metastases, when these tumors are co-cultured with adipocytes, they tend to do a lot better. They seem to thrive on that, sort of the fat cell. So I wonder if is this just a manifestation of metabolic alterations that are happening because of your high fat diet and chow?
Yeah, that's a great question. We have right now actually metabolomics samples that are out pending. Looking forward to it back hopefully next week. I think that our germ-free model shows at least that all of those mice in both groups were the same weight, exposed to the same diet. So there is at least some signal from the microbiome isolated in that model. However, I do think it's more complex than that. I think there would be crosstalk between a paracrine signaling for those adipocytes, and then whether it's the bacteria themselves producing the metabolite that would impact tumor cells or vice versa. We don't have data to say exactly what signaling is altered with that high fat diet, but that's really the next thing we're looking at.
That's great
Yeah,
work. Was someone asking?
No, wait.
I was gonna say, I think that's the first time I've heard the word cancer cell enema. Okay, I'll have to remember that one. Cancer cell enema was given. But I think this is a great project because it really asks a very fundamental question, which is what can the patient do right beforehand? What can they do? They all ask us this, right? What can I do while I'm waiting for my surgery? Can I change my diet now, or is it too late? And what you've shown is it's not too late. You can change your diet right before surgery and make a difference. And while I sort of agree with what you two said about the model may not be perfect one-to-one, the point is it shows that, in an implantation model, invasion model, you're making a difference. I mean, it may not be the perfect model for this particular clinical question, maybe more common, but it sort of answers the question, which is, do you get more tumor invasion? Do you get more implantation, more survival of the tumor cells in a wounded environment, which, all surgical areas are, when you have shedded cells? I do think it answers that question. I was more curious what you found. The other group of mice actually had the opposite finding, which, you
Yes.
didn't delve into too much, but I thought that was almost equally interesting.
Yes.
You don't have to show the slide, but that kind of caught my attention because that sounds like it's a signal that should also be researched.
Definitely. And that's
It was protective for them, actually.
Yes.
Sorry.
Yeah. So that was, I think, one of the more interesting things is that these two organoids, which are quite similar, the process of giving the NMS part of the just have the different genes that have the opposite response to the high fat diet. The kind of work that we're doing with the KPN tumorids, we plan on repeating that with the KPN tumorids just for completeness when we go to some of this from.
Because I was going to either change my high fat diet, but then I said, I don't know what kind of tumor I'm going to grow, so I can just stay on it because it might be protective in certain cases. I'm not going to change it. I'll stay.
Great. Thank you so much, Ryan. I always find it interesting to listen to clinicians try to reason what they observe clinically to what experimentation can produce in terms of advancing our understanding of a complex disease problem. And I think that's what we all struggle with. What's interesting for me is listening to the Taraga on Keith's presentation, showing that there are things in the bloodstream at the level of genes that you can detect before imaging produces something. And so when we see a lot of liver metastases, we assume that the pelvis is sterile. But we only know that the pelvis is sterile based on imaging alone. It's not as if when you do a liver resection, you go diving into the pelvis and sample every node and send it off for detection. And most of those patients have received neoadjuvant or adjuvant chemo-RT. So you may be cleaning up what was in the pelvis or around the anastomosis. And then the last thing to manifest itself is this liver, which you now say, oh, it's an oligomet. There's nothing in the pelvis. It couldn't have come through the anastomosis. But this opens up the possibility, at least, that to Dr. Parenke's point, that there's some things that can be manipulated beforehand that might have, a benefit later on. Thank you, everybody, for your participation in those excellent comments. We're now going to have our last presentation before the break, which is by Dr. Jason Chow, who is a medical student from the University of Chicago, who has spent some time in my laboratory, both in the SRP program and now continuing along. He's been working with Bobby Keski and Olga Sabarina on the model that they're about to publish, and the model that serves for Bobby to get his PhD through the Committee on Immunology, and that is on survival from Serratia marcescens peritonitis. Depends on competition for aryl hydrocarbon receptor signaling in macrophages between gut microbiota indole metabolites and pathogen-derived signaling molecules. Jason.
Thank you, Dr. Alberti, and I want to thank everyone for being here today, for allowing for this opportunity to present. So as you know, infection is a leading cause of mortality, especially for surgical patients. And the response to infection is complex. Now, many studies of infection typically involve identifying a gene of interest and then knocking it out in mice, and then infecting those mice and showing that there's a difference in survival between those groups. Now, what's interesting is if you hone in on any particular group, for example, if we hone in on this wild type group, right? These individuals are going to have the same genetics. They are living in the same environment, and they're exposed to the same pathogen. And yet, what you see is that some of them will die, but some of them will live. And the question is, why? Well, in our lab, we were wondering whether or not differences in the gut microbiome, which varies by individual, could potentially help explain this kind of within group survival difference, specifically through the metabolites that they produce. Now, this is a concept that has been, that is not novel. It's been shown that different metabolites, such as short chain fatty acids, do have potential to regulate the immune system and to modify the inflammatory process. So in order to study this question, our methods, we had to develop a model. And so we developed a model of monobacterial peritonitis using a 50% lethal dose of Serratia marcescens, which is a gram-negative bacteria. And in this model, we can use core body temperatures to reliably predict the survival outcome of a mouse. Now, using this model, our goal was to determine if gut microbial metabolites could distinguish between the surviving and the non-surviving mice. And we approached this question from two different angles. We thought that if metabolites could indeed influence survival, that either it could influence the host immune response to overcome the pathogen, or it could directly affect the pathogen itself. So the first question we had was, okay, well, is there actually a difference in the level of metabolites between the survivors and the non-survivors? What we found is that tryptophan metabolites are elevated both in the gut and in the peritoneum of the surviving mice. And specifically, these are indoles, which are produced only by the gut microbiome. And indoles are known to activate a protein called the aryl hydrocarbon receptor, or AHR. So these indoles bind to AHR, and then AHR translocates to the nucleus, and then drives the expression of a number of immune regulatory genes. So the idea from here is that tryptophan metabolites make their way into the peritoneum, and they can activate AHR in a number of immune cells. And so two questions that arise from this are, one, what immune cells do we think are involved? And two, do these metabolites activate AHR in them? Well, when we think about the first responders to a very acute model of bacterial peritonitis, we think about the peritoneal macrophages. So what we found is that when we deplete the peritoneum of macrophages, that all of the mice die, indicating that the macrophages are an important player in our model of infection. Moreover, we found that there's actually phenotypic differences between the macrophages of surviving mice versus the non-surviving mice. Specifically, there's an elevation in expression of arginase 1, which is kind of a marker of an M2 kind of anti-inflammatory macrophage phenotype. So seeing that macrophages were an important part to this story, we want to know, okay, well, do these metabolites activate AHR in them? And to do this, essentially, we combine the four most highly elevated metabolites that we detected, and then we expose them in vitro to macrophages that are derived from bone marrow of mice. And we use downstream gene expression as a kind of readout for HR activation. And what we found is that macrophages that had been exposed to the indoles did in fact have increased AHR signaling, and that this signaling was abrogated when we added in a selective AHR inhibitor. So now that we established that these metabolites have the ability to activate AHR in macrophages, we wanted to know some of the functional consequences of that activation. And so the first experiment we did was we actually incubated macrophages with serratia, and then we lysed the macrophages and counted the number of serratia that were still alive inside. And what we found is that macrophages that had been exposed to these metabolites were more efficient at clearing the bacteria itself. In addition, these metabolites increase the expression of genes that are consistent with what we found in vivo. For example, arginase 1, IL-10, again, are associated with an M2 phenotype of macrophage and are thought to prevent excess inflammation. Importantly, both of these effects were no longer observed when we added in an AHR inhibitor. So seeing these effects in vitro, we wanted to know whether or not AHR signaling is important in vivo, i.e. does it affect our outcome? So we found that when you selectively inhibit AHR, that all of the mice die in our model, indicating that the AHR signaling is important in this model. More specifically, when we do a selective knockout of HR only in macrophages, that the mice have increased mortality, which is consistent with what we're thinking right now. So at this point, we've kind of seen that these metabolites have the potential to activate HR in macrophages. We also wanted to know, do they affect the pathogen itself? So how do they affect serratia? And essentially, when we what we found is that these metabolites in a concentration-dependent manner delays the growth of Serratia marcescens, and it also inhibits their ability to form biofilms, which is one of Serratia's virulence properties. As a better readout of virulence, however, we actually used a small animal model of Galeria melanella, where we inject Serratia along with the indoles into the Galeria. And what we found is that administering the indoles actually delayed mortality. And that this effect was seen for a number of metabolites that we tested. So given the importance we think AHR might have in overcoming this infection, we wanted to know whether or not the pathogen, actually Serratia, played a kind of counter role. Could it affect AHR signaling as well? And so For this, we took a cell line that luminesces when AHR signaling is active. And what we found is that when you add in the supername of serratia, it actually has the ability to kind of attenuate or suppress AHR signaling that's induced by these metabolites. Now, this is a very preliminary kind of experiment, but it's very exciting, and I think it adds a little bit of complexity to the current paradigm. So we've explored the effects of metabolites on the host immune system as well as on the pathogen. Our final question was to ask, does administering indoles actually improve survival in our model? And so we returned to our mouse model. This time we found that in the mice who had been administered indoles along with the serratia, that they had improved survival compared to the vehicle control. In addition, these mice had higher, healthier core body temperatures and displayed less severe signs of clinical infection. And then finally, when we sample the peritoneal fluid in these mice, we find that the treated mice have reduced bacterial load in the peritoneum. So we started off with a question about within group mortality. And we wanted to know whether or not the gut microbiome, specifically the metabolites that they produce, could help explain this difference in mortality. And what we found is that there's an elevation in tryptophan metabolites that have the potential to inhibit the growth and the virulence of Serratia marcescens. In addition, these indole metabolites have the ability to activate AHR in macrophages, which enhances bacterial clearance and may prevent excess inflammation. In contrast, Serratia may actually actively inhibit AHR signaling and subvert host immunity through that mechanism. And then finally, when we administer these metabolites in 2 different animal models, we find that it improves survival or it delays mortality. And that is my presentation. I wanted to thank everyone for this opportunity again, to the Department of Surgery, and of course to all the lab members in the Alberta lab that made this possible. Thank you very much, and I'm open to any questions.
Great job, Jason. Thank you very much. Are there any questions? Dr. Matthews looks, or Dr. Prangi?
Oh,
Looks
great
like somebody-
presentation. I can't believe you're a medical student. You can present like that. Fantastic job. So it was very complex in the sense that, you know, even the bacteria itself is affecting the pathway. So what, I guess, where do you think are the actionable steps? Is it the giving of the indole? Is that something that can be given currently to patients? Is it an orally available agent? Is it something that's not toxic? I don't know much about that.
Yeah, so I think it would be far and away to the point where we could actually take actionable items, I think, in terms of, you know, elevating level of immune enhancing metabolites for patients. I think the thrust of this project is mostly looking into what other factors can explain the individual response to infection. When these, like, you know, Ryan had an amazing presentation on different genetics and that interaction, but we're wondering, you know, if they're genetically similar, right, which doesn't happen in the real world. But say, for example, if, you know, take out genetics, take out environment, they live in the same cages, right, these mice. Take out the pathogen they're exposed to, but because we use a monobacterial model, whereas in reality it's going to be polymicrobial, it's going to, you have no idea what's actually infecting your patient. So we take out those variables and we still find differences, and we think those differences are coming from the gut microbiome. And so I think this is an important study because it looks into one potential mechanism by which the gut microbiome influences outcome. Because I think at this point we acknowledge that the gut microbiome is important in terms of you know, survival, infection, cancer, but this I think this study is more about looking into what's one way that it actually influences that outcome. So we're diving into the sort of the
Deep.
metabolomics portion of that question. So yeah, at this point, I would not recommend, like injecting
A
these metabolites.
particular medicine.
Yeah, but
medical
I
route.
think being aware of it is helpful, because the way, in the hospital, we do affect our patients' microbiomes all the time, whether or not it's changes in diet, it's antibiotics, bowel prep. So I think it's there's still a lot of work to be done. But I honestly, I'm sure you all know Dr. Kasky. He's great. He's kind of the one who helped bring this.
Well,
bring
I
this
love
going.
how you say that with such definitiveness, that it's known that the microbiome does all these things. And I think it's really the work of Dr. Alberti and people like Dr. Kesky who've made that obvious. I don't think that was that obvious that long ago. I love it how you say it so definitively. That is
We
great.
say it around here. I don't think
Yeah.
it's known outside. That was a great presentation. I was really struck by one of the findings, which was the impact on body temperature. So it's been known for a very long time that body temperature affects white cell killing. If you give Tylenol in models of experimental models of sepsis, you increase mortality actually, and it's probably a body temperature related phenomenon. It's why there's been an age-old debate, which everybody wants to ignore, about whether giving Tylenol to postoperative patients is a safe thing or not, or whether it actually increases septic complications. So this whole issue of antipyresis and outcome from sepsis, which is a really important surgical question. So do you know whether your indole compound just independently affects mouse body temperature? Has that ever been looked at? And could some of these effects be entirely explained based on body temperature?
Yeah, that is a really interesting question. So it has not been looked into, but I know what I'm going to do, next month. But, no, I think that's a really interesting point. I would say indoles broadly have a number of functions. Right now, we're just looking into one pathway. There's a lot. Indoles are used as signaling metabolites between species of the gut microbiome, not just through the host, through AHR. So there's actually, I'm sure there's actually quite a few other functions that it has, and it affecting temperature is a very interesting idea, and I could see how that could influence.
If you look if you actually look up antipyresis and the outcomes from it, you find a literature that people have ignored for decades, showing the risk of changing body temperature. It's no accident that sick lizards that are poikilotherms go to the sun. They move to elevate their body temperature as an adaptive response to try to survive sepsis. And when we experimentally, we have, in humans, when we see patients with fevers, we instinctively knee-jerk, cooling blankets and Tylenol and everything to try to get their fever down, when it's in fact an adaptive response. And just a degree or two of temperature can dramatically affect white cell killing. So it's always something to keep in the back of your mind, whether there's some other thing that it's doing in addition to what you're seeing here. Great work. Any other questions?
I think.
I not catch it.
Sorry.
There's an audience question.
Who's speaking?
Yeah.
Identify yourself, please.
It's Kathleen Cow.
Okay.
So I don't have an MD and I never took biology, so I have a naive question. So if you say that the microbiome is unique and individual to everybody, is there a way to then sequence or look into that beforehand to kind of see what is the profile of the microbiome? And then perhaps then eventually figure out if there is some kind of pattern as to overall, like what features can lead to survival and which don't?
Yeah, definitely. So there are ways to do that. I would say because the microbiome, it can fluctuate even in the same person. I think it might be a little difficult to say have a patient come in 6 weeks before, and then, I guess assuming they're on the same diet, nothing changes, potentially. So that is a very tricky aspect, but it's something, but in terms of like predictive value, that's actually one of our like next lines of work is to see, even pre-infection, can we like measure these levels of these metabolites? Can we look at the gut microbiota composition? And then see whether or not track those mice and see what happens to them. I will say, sort of interestingly, let's see, this is some work that is not in the presentation, but essentially that there aren't any. It doesn't seem like there's actually drastic changes in the composition of the gut microbiome between the surviving and the non-surviving mice, but it's more kind of on the metabolites that are produced. But I do think that's a really interesting question, and it is something that we're hoping to do next.
Great.
Just have a bad arm, I can't throw it.
So just 1 million question, just to answer. Just 1 million question from naive girl. Yeah, we run actually those experiments, and we already shown that we cannot predict it based on the composition pre-IP because the composition is the same. Metabolites becomes different. Metabolism is much more flexible compared to composition. And I think this is the rule. So we continue those results, those experiments right now. So maybe next time we can answer your question.
Jason, can I ask one quick question? I know you said this is sort of a balance between attenuating the immune response and killing, but it sounds like it was doing both. Just curious how you're balancing that seeming contradiction.
Yeah, so that's really interesting. So a couple of things to mention is I would say, first of all, when we measure like gene expression, that's kind of a snapshot. Whereas in reality, infection's taking course over multiple hours. And that's just something that we don't know because, right, we're just taking a single snapshot and we're saying at this point, there's kind of been like an anti-inflammatory kind of phenotype. So I would say that, the story is much more complicated than that. In addition, I think it's all about having a homeostatic level of inflammation. So you don't want too much, but you don't want too little. And I think oftentimes in cases of infection, sometimes your immune system goes a little bit overboard and ends up harming your own body. And that can lead to ultimate, not surviving. So I would say you want to help achieve like a very fine level of that in your body's immune response, and achieving that balance can be potentially done through the metabolic signaling that's occurring. Sorry, I don't know if
That's good.
It's great.
Thank you.
Okay.
Okay. Wonderful. Thank you very much, Jason. Nice job. Good questions. Thank you all for participating and being engaged in this. We're going to take a 15-minute break. Please come back right at 2 p.m. We have some outstanding presentations coming, especially from our residents who have spent time in the lab, and of course, Dr. Prangi at the end that Dr. Matthews will introduce. So please hang in there, and we'll see you in 15 minutes.
Thank you.